Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today I will discuss some multiple choice questions with you people. And I have taken this MCQs from United States of Medical Licenses Examination that you may know USMLE. So the MCQs are related to microbiology, and I have taken out the bacteriology section, or in simple basic bacteriology. So I'm planning to make a video series of that is related to microbiology, and I will discuss different MCQs that has appeared in the USMLE examination. Okay, so you can check yourself that how much is your knowledge, because these MCQs are very much authentic and reliable. Mostly all the MCQs you will see in a minute that all are very much conceptual. There is no straightforward MCQs, means that what is the size of a bacteria, what is the size of a cell wall. So it does not consist of the straightforward question. All the questions are conceptual. So you can pause the video, you can take the correct answer, and then you can compare your answer with my solution. Okay, I will discuss all the MCQs, the parts of the MCQ, that why these are wrong and why the correct answer is, and what is the reason that I have chosen the correct answer. Okay, if you have any problem, so in my answer, so you can comment me in the comment box. It can also boost up my knowledge, and I'm planning to make videos with respect to different topics, that that is basic bacteriology, clinical bacteriology, virology. Okay, so in today's video, I will discuss 15 MCQs, and in the later videos, I will also take a specific sum of MCQs. Okay, so let's start with the MCQs. Okay, so as I have told you, so that I have taken it from the basic bacteriology. The first question is that each of the following statements concerning the surface structures of bacteria is correct except. Okay, I have told you that all the MCQs are very much conceptual. Okay, so we are having four statements, and three are correct and one is wrong. Okay, about the surface structure of the bacteria. Okay, so the first one is that pili mediate the interaction of bacteria with mucosal epithelium. So it is correct, because pili is present on the surface of the bacteria, and it helps in the attachment to the mucosal epithelial. So in simple, it helps in the interaction. Okay, so this is not our answer. The second is the polysaccharide capsules retard phagocytosis. As we know that a capsule is a special type of covering that retard phagocytosis, means that it can slip away from the phagocytes, from the neutrophils and the macrophages, and the bacteria cannot be engulfed easily. Okay, so it is also correct. The C is that both gram positive bacteria, okay, both gram negative rods and cocci. Okay, wait a minute. Okay, so both gram negative rods and cocci have lipopolysaccharide in their cell wall. So it is also correct statement that only gram negative rods, either it is cocci or rod shapes, they are having LPS, okay, lipopolysaccharide in their cell wall. The fourth is that bacterial flagella are non-antigenic in humans because they closely resemble human flagella in chemical composition. Okay, so in simple, it is not correct. Okay, because the bacterial flagella is not the same as the human flagella. Okay, so that's why it is antigenic. So our answer is D. Okay, because each of the following statement concerning the surface structure of bacteria is correct, and so the D is not correct. Okay, second is that each of the following statement concerning peptidoglycan layer is correct except. So three statements are correct related to peptidoglycan layer, and the one is wrong. So we have to choose the wrong one. Okay, So peptidoglycan layer, it has a backbone composed of alternating unit of uramic acid and acetylglucosamine. We all know that the alternating units are name and name. Okay, so that's why this is not the option. B is that cross links between the tetrapeptide involve DNAE. Okay, so this is correct that we are having a pentapeptide or tetrapeptide chains, and it is connected. The DNAE is connected to the lysine. Okay, you can check it in the peptidoglycan layer. Okay, so it is also not the correct answer. It is thinner in gram negative, gram positive than in gram negative cell. So now this is our answer. Means that the peptidoglycan layer is thicker in gram positive and thinner in gram negative. So they just rotate the question. Okay. So this is our answer. Now it can be degraded by lysozymes. So yes, peptidoglycan layer can be degraded by lysozyme. Each of the following statement concerning bacterial spore is correct except, okay, so 3 MCQs are correct related to endospores. So just look here, their survival ability is based on their enhanced metabolic activity. Okay, so keep this thing in mind that in endospore there is no, there is very little amount of metabolic activity. Okay, so our answer is correct. There is no enhanced metabolic activity in the endospore. Okay, so this is our correct answer. Just look here, these 3 statements about the endospores are correct. They are produced by gram positive rods, and they are produced by bacilli. Okay, and bacillus bacteria. They can be killed by autoclaving. Yes, it is correct, and they are formed primarily by nutrients are limited. Okay, so we all know that when there is nutrient limitation, so endospores are formed. Okay, in order to just avoid that condition. Now, which of the following statement is the most accurate comparison of human bacterial and fungal cells? Okay, so we are having 3 cells and we have to choose the most accurate comparison. Okay, so human cells undergo mitosis, whereas neither bacteria nor fungi do. So it is not the correct answer because human undergo mitosis and fungi also undergo mitosis. Okay, so that's why it is not the correct answer. Now, B, human and fungal cell have a similar cell wall in contrast to bacteria. So this is also not correct because human does not have cell wall. So this is not the correct comparison. Now, human and bacterial cell have two plasmid and fungal cell do not. So human does not consist of plasmid. Whether bacterial cell have plasmid, but human do not have. So this is not the correct comparison. D is the human and fungal cell have similar ribosome. Yes, it is correct because human cells and fungal cell have 80s ribosome and bacterial ribosomes are different because they are 70s. Okay, so this is our correct answer. Now, fifth is that which of the statement is the most accurate regarding the drug depicted in the diagram. So just look here to the drug. This is an antibiotic and basically it is an aminoglycoside. Just look here, these are the sugar rings and the NH2 is the amine group. So this is basically aminoglycoside, and aminoglycoside is basically protein synthesis inhibitor. Okay, so now just look here to the options. It inhibits DNA synthesis. No, it is involved in the protein synthesis inhibitor. So this is not our best answer. It is bacteriostatic. So keep this thing in mind that aminoglycosides are bactericidal. These antibiotics are not bacteriostatic. Okay, so that's why this is also wrong. It binds to 30S ribosome. So aminoglycoside binds to 30S ribosome. We all know. So this is the correct answer. It prevents formation of folic acid. No, it is. These are other antibiotics. Okay, these are not aminoglycoside. Now, sixth is the each of the following statement regarding the selective action of antibiotic on bacteria is correct, except, okay, we are having 4 statements and that are basically regarding the mechanism of action of antibiotic, but one is wrong. Okay, we have to check on the wrong. Okay, so chloramphenicol affect the large subunit of the bacterial ribosome. Okay, chloramphenicol is an antibiotic, and we all know that it is a protein synthesis inhibitor, and it binds to the 50S ribosomal subunit. Okay, so and it does not affect, it does not bind to the human ribosome. So this is not the answer of it. basically it is the correct mechanism of action. Isoniazid affect the DNA polymerase of bacteria, but not that of human cell. So now isoniazides are basically antibiotic that are involved in the destruction of the mycolic acid synthesis. Basically, they block the mycolic acid synthesis of the Mycobacterium tuberculosis. Okay, so when there is a patient and he is witnessing TB, so basically isoniazides are recommended because they kill the bacteria in a sense that they inhibit the synthesis of the mycolic acid. Okay, so this is our answer. This is not the correct mechanism of action. Sulfonamides, they stops folic acid synthesis. Okay, affect folic acid synthesis of bacteria. This is the correct mechanism of action, and penicillin affect the bacteria rather than human cell because human cell do not have cell wall. Okay, so this is also correct. Now, 7. Each of the following statement concerning endotoxin is correct except. Okay. Now you have to make sure that your knowledge about endotoxin are quite well. Okay. So they are less toxic than exotoxin. Okay, they are less toxic than exotoxins. Okay, so yes, the endotoxins are less toxic than exotoxins. Exotoxins are much more toxic than endotoxin. So this is not our answer. They are more reliable. So sorry, they are more stable on heating than exotoxin. Yes, endotoxins are basically more heat stable than exotoxin. They bind to specific cell receptor, whereas exotoxin do not. So if it is endotoxin or exotoxin, they have to bind to the specific cell receptor. So this is our answer. And they are part of bacteria cell wall. So yes, endotoxins are part of the gram-negative cell membrane. Okay, and exotoxins are just released into the environment. So this is our answer. C is our best answer. The main host defense against bacterial exotoxin is, so these all are the host defense mechanism against exotoxin, but one is the most accurate one. So we have to choose the most accurate one. So activated macrophages secrete proteases. So exotoxins, basically exotoxins does not activate the macrophages. Means that the best answer is that basically exotoxin, when there are exotoxins, so our immune system manufacture IgG and IgM. In a sense, they synthesize antibodies in order to degrade them, in order to kill them. Okay, so this is our correct answer. Helper T cells are also activated, and the cell receptor also give response to the toxin. But the best defense against exotoxins are the IgG and IgM antibodies production. Which one of the following process involve a sex pilus? Okay, so basically, what is a sex pilus? Basically, bacteria consist of the cell. There is a special type of organelles that are known as pili proteins. Okay, or in simple pili. So pili are a special type of protein that help in the attachment. Also, they are used in the transfer of genes from one bacteria to other bacteria. They just make bonding with the help of that sex pilus, and after that they release they transfer their genes across themselves. Okay, so now just look here. Transduction of a chromosomal gene? No. In transduction, basically bacteriophage is involved. Okay, so this is not the correct answer. Transposition of mobile genetic elements? No. Transposition is carried out by transposomes. Okay, now C. Integration of temperate bacteriophage? No. Here is bacteriophage involved, not sex pilus. The last is the conjugation. So basically, conjugation is the process in which sex pilus is involved. They elongate their pili protein and they just stuck into the other bacteria, and the gene of interest are in the form of plasmid are interchanged between the bacteria. Now, strength is that each of the following statement concerning the normal flora is correct except. Now, this question is about the normal flora. So, you have to know about the normal flora, okay? So, okay. The most common organism found on the skin is Staphylococcus epidermidis, okay? Yes, Staphylococcus epidermidis is a special type of bacteria, gram-positive bacteria that is present in a high amount on the skin. Okay, E. coli is a permanent member of normal flora of the throat. No, this is not correct. E. coli is mostly present in the large intestine, and these two are also presenting the best location. Bacteroides fragilis is present in the colon, and Staphylococcus aureus is present in the nose. Okay. Now, 11. Each of the following statement concerning the mechanism of action of antimicrobial drug is correct except. Okay. Now we have to choose the best. We have to choose the mechanism that is not of the antibiotic. So vancomycin acts by binding peptidoglycan synthesis, inhibiting peptidoglycan. Yes, vancomycins are cell wall inhibitors. Quinolones such as ciprofloxacin acts by inhibiting the DNA gyrase. Yes, quinolones are basically DNA synthesis inhibitors. They bind to the DNA gyrase and they just stop the production of the DNA replication. Okay, and you can check this mechanism of action on my YouTube channel. Erythromycin is a bactericidal drug. Okay, erythromycin is a bactericidal drug that disrupts cell membrane by a detergent-like action. Now, this is not the correct function because erythromycin are basically protein synthesis inhibitors and they are bacteriostatic. They are not bactericidal, okay? And they are protein synthesis inhibitors. They are not cell membrane inhibitors. Basically, polymyxins are cell membrane inhibitors, okay? They just disrupt the cell membrane. So this is our correct answer. And amino glycoside, the correct option is shown, okay? Now, each of the following statement concerning the resistance of bacteria to antimicrobial drug is correct except. Okay, now here is the resistance. This is a little bit tricky one. You have to know about the resistance, okay? So resistance to chloramphenicol is known to be due to an enzyme that acetylates the drug. Yes, chloramphenicol is a special type of antibiotic and bacteria can show resistance to it. And scientists have found that the chloramphenicol is modified by the bacteria and they just acetylate the drug. So then the drug cannot no longer act correctly, function correctly. Okay, so that's why this is the correct. Now, second is the resistance to penicillin is known to be due to reduced affinity of the transpeptidase. Basically, transpeptidase are penicillin binding proteins, and these proteins are basically involved in the cell wall biosynthesis. So now bacteria show resistance in a sense that they modified their transpeptidase enzyme. So the penicillin can no longer bind to it. They reduce their affinity. So yes, this is also a type of resistance. We have to choose the wrong one, okay? Now here is the resistance to penicillin is known to be due to cleavage by beta-lactamase. Yes, penicillin have beta-lactam rings and bacteria synthesize beta-lactamase enzymes and they degrade, just destroy the beta-lactam ring. And the fourth is the resistance to tetracyclines is known to be due to an enzyme that hydrolyzes the ester linkage. Okay, no, tetracycline does not have any ester linkage. Okay, so this is the correct answer. Now, each of the following choices the most important function of antibody in host defense against bacteria. Now, here we have to look to the best function of the antibody. So, activation of the lysozyme that degrade the cell wall, no. Acceleration of the proteolysis of the exotoxin, no. This is not the function of the antibody. Facilitation of the phagocytosis, yes. This is the correct option because it enhances opsonization, okay? So this is the correct answer. Now, inhibition of the protein synthesis, yeah, no, that does not involve this function. 14, the second last is that which of the following event is most likely to be due to bacterial conjugation. As I have told you that in bacterial conjugation, you have to look to the antibiotic resistance genes. Okay, when mostly in conjugation, plasmids are transferred between bacteria, and the transfer of the plasmids, basically plasmid encodes resistance genes to an antibiotic. Okay, so mostly resistance is transferred with the help of conjugation. So a strain of Corynebacterium diphtheria produce a toxin encoded by prophage. No, this is basically transduction. B. A strain of Pseudomonas aeruginosa produce beta-lactamases encoded by plasmid similar to plasmid of another gram-negative bacteria. So yes, the beta-lactamases are transferred by the plasmid with the help of the conjugation. So as I have told you, these two are also not correct because they are not involved in the uh, resistance. Okay. 15, last one is that which of the following best describe the mode of action of endotoxins. Okay. So endotoxins are basically uh, degrade lecithin in cell membrane. No, this is the function of the exotoxin. They degrade lecithin. Okay. B is that it inactivates elongation factor 2. Basically, elongation factor is inactivated by exotoxins. Block release of acetylcholine? No. It is a function of the exotoxin. The D function is correct because it causes the release of tumor necrosis factor. Okay. So this is the correct option. So that's all for today. In the next video, I will discuss the remaining MCQs or that is related to basic bacteriology. If you have any question, you can comment me. Okay, I will make more videos related to these tests, and then you can check yourself. Thank you for today. Thank you very much. God bless you.